First, many thanks for inviting me and offering me the opportunity to discuss our work. Some of the work is actually still unpublished and really just out of the oven, so to speak. I have a long-time fascination with social interaction. I think it's very obvious for us humans, particularly meeting in a room like this. We really thrive when we are in group, when we are together with other human beings. It's very clear also that our brain is constantly ruminating about the people we like, the people we hate. Social interaction also is seen here, and I think this very beautiful sculpture of Camille Claudel. Our social interaction really inspires some of the most beautiful art, both to show joy or to show pain caused by successful or unsuccessful social interaction. So I think it's fair to say that the human brain literally is geared towards social interaction. There's something just fundamentally important about social interaction, and I think it's really important to try to understand why these social interactions are so important and what are the brain area and circuit that govern those behaviors. And also, very importantly, social interaction are probably the first set of behaviors that are affected by a number of mental disorders, including depression, schizophrenia, autism, and quite a number of other debilitating mental illnesses. And so how to study this? Well, obviously we can't directly work on human as an experimental animal, but it's also the case that animals require to interact with their conspecific in order to thrive, in order to reproduce. They need to defend their territory, they need to defend and nurture their young. So there are quite a number of at least elements of instinctive social interaction that can be found in animals. And so the idea is that at least at the level of the brain, there will be some conserved element of social circuit in the brain of large number of animal species. And what will likely differ, however, are the specific signal that trigger specific social interaction. Those signals are very likely to be extremely species-specific, such that one set of animals are entrained and triggered to interact with a specific set of other animals. And so to illustrate this, I would like to show you a couple of what I think are very interesting example. So in birds, for example, this particular bird species called the American flicker, in this species, the male and the female recognize each other by virtue of one single trait. And that is the presence of this black mustache here on the face of a male. So if an American flicker has a black mustache, it's a male. If it doesn't have a mustache, it's not a male. So if you mask this mustache, if you paint over it, then the other male will try to mate with this animal because they think it's a female. And similarly, if you paint a black mustache on the face of this female, then the other male are going to attack this female because they assume it's a male. So in other words, I have a mustache, I'm a male. I don't have a mustache, I'm a female. So visual cues are very important for birds. Similarly, in rodents, in many species of rodents, a specific set of chemosensory cues, chemical cues called pheromones, are essential to drive social behavior in these species, in rats, in mice, and a number of other rodent species. Humans are sensitive to other type of cues, mainly visual and auditory cues, and those cues really are sufficient to trigger exactly the same type of urges that pheromones and visual cues trigger in birds and rodents. And these explain the enormous success of the pornography industry that is clearly zooming in on that particular type of circuit in the human brain. So our animal model is the mouse, and the mouse, as Louis introduced, like many terrestrial vertebrates, terrestrial mammals, but actually not humans, are very sensitive to social scent. And what you see here is called the Flemming response, which is this very specific way of animal to capture social scent in the environment. And what they are trying to do here is to capture chemicals that will access a particular set of neurons in this organ called the voronasal organ. And these neurons then send projection to another set of neurons in the accessory olfactory bulb, and then in what is called the voronasal amygdala. So these constitute what is called the voronasal system, which again, in many mammals, is specialized in detecting these social cues called pheromones. And I must say, humans do not have a functional voronasal system. The other set of olfactory neurons are the neurons of the main olfactory system. These neurons are sensitive to volatile cues that enter the nasal cavity during breathing, and these signals are sent through a very different circuit to the main olfactory bulb and then to multiple areas that constitute the primary olfactory cortex. Now, these two circuits rely on different set of signaling molecules. In the voronasal system, we and other group identified two distinct family of voronasal receptors that detect these voronasal cues. And Richard Axel and Linda Buck, as Louis introduced, identified this very large family of olfactory receptor genes. Now, interestingly, these receptors rely on very different ion channels to transduce the chemical signal into electrical activity. In the voronasal organ is expressed this ion channel called TRPC2 that is essential for VNO signaling, and in the olfactory system, the olfactory cyclic nucleotide gated channel is essential for olfactory transduction. And because of the difference between these two signaling pathways, many years ago, we were able to assess the function of each of these systems by deleting each of these signaling molecules. So in mutant mice, for the TRPC2 channel, these animals are unable to detect cues through the voronasal system, so they are VNO blind, if you wish. And in contrast, if you delete the cyclic nucleotide gated channel, these animals do not have a functional olfactory system. And at the time, what we expected is that animals that are mutant for the TRPC2 channel would not detect pheromones and therefore would be unable to mate. And surprisingly, this is absolutely not the result what we found. What we found is that animals without a voronasal system were perfectly able to mate, but animals that were ablated for their sense of smell were not able to mate. So in other words, it seems like the sense of smell, or at least a subset of neurons in the olfactory epithelium, was essential to trigger mating behavior. And the voronasal organ is doing something else. So what does it do? Well, what we found is that animals that were deprived of voronasal function were unable to discriminate between males and females, which is that when a male was encountering another male, instead of attacking that intruder, it was actually attempting to mate with that other male. And it would also mate with a female. And if you put male and a female together in a cage, then the voronasal mutant would mate with one, and then the other one clearly would not be able to tell apart what would be a male and what would be a female. So again, the idea is that the voronasal system is here to identify gender-specific cues, and a male detecting a female would lead to mating behavior, and a male detecting another male would inhibit mating behavior and instead trigger attack. So to some extent, exactly the opposite that what we had expected. We found that the voronasal system is here to block mating behavior when the wrong type of cues is encountered. And interestingly, this theme has been expanded recently by other groups. So for example, Steve Lieberles showed that the voronasal organ is essential in blocking the mating behavior of adult males towards juvenile mice. And Niraj Shah similarly showed that the VNO is able to block the mating of male mice with other type of rodents. So somehow this seemed to be the role of the voronasal system, at least one of the roles. Now, all these studies that I've just described were done in male mice. And so in parallel, or a little bit later, we now address the role of the voronasal system in female mice. And here the results were absolutely astonishing. And again, absolutely not what we had expected. So first, I must say, people in the lab were very reluctant to look at female behavior because very few female-specific behavior have been described in the literature. But finally, a very talented postdoc, Tali Kimchi, decided to look at something very simple, just sexual receptivity in female mice. And so what she did in this assay is to look at a female mutant for the TRPC2 channel, so an animal that doesn't have any functional VNO, that's the animal here in gray, and put this in presence of a male, shown here in black. And as you will see, the behavior of this female is very surprising. Again, the animal in gray is a female. And so you can see this female is actually behaving exactly like a male would behave, trying to mount these other males. And actually, this female would have the same behavior with a female as well. So somehow, this female has the social behavior of a male. Not only the social behavior, but it also emits ultrasounds exactly like a male would emit. It also displays reduced maternal behavior. And to prove that this behavior was not due to the miswiring of the brain during development, we removed the voronasal organ in the adults. So we let a female develop and then surgically ablated the VNO and tested the behavior of this female and found exactly the same behavioral phenotype. And this, as well, we found that these animals have normal level of steroids. And so this was telling us something very different. It told us that somehow the voronasal system in the mouse is there to control the sex specificity of behavior, the gender specificity of the behavior of the other animal by repressing male-specific behavior and triggering female-specific behavior. So if you remove the voronasal organ, now suddenly some ectopic male behavior appear and some amount of female-specific behavior is not displayed. And so this was very surprising because it really seemed to contradict some well-established ideas on how the male and the female brain develop. So in a little cartoon, what we found is that the female brain seemed to have behavior circuits that control male-specific behavior as well as controlling female-specific behavior. So the voronasal system would be these arrows here that are able to trigger female-specific behavior, shown here in red, and inhibit male-specific behavior, shown here in blue. So if you remove the voronasal system, suddenly you see emerging this ectopic male-specific behavior, and you see defect in female-specific behavior. So this is quite different from the classical model on how brain sexual differentiation occurs that has postulated now for almost 50 years that the way the male and the female brain emerge is that right at birth, young males have a peak of testosterone, and this peak of testosterone leads to the organization of a specific set of male behavior circuit. And the female that do not have this peak of testosterone develop instead a different type of behavior. Female-specific behavior. But this model here could absolutely not account the type of phenotype that we observed in the TRPC2 female mice. Now, In retrospect, this result, the result we obtain and the model that we propose is actually not something really new. It has been shown already in the 20s and the 30s by some very distinguished ethologists that sex reversal in the mating pattern of males and females were observed in almost all mammalian species observed, which is that in all these mammalian species, there is always sometimes a male behaving like a female, and females occasionally behaving like males. And it's interesting to read this text and to see, you know, at the time people didn't talk about secret, but they said there must exist the innate organization essential to the mediation of the mating pattern of either sex. So the idea is that maybe there's something intrinsically bisexual in at least some portion of the brain, and the role of the hormonal system, or maybe other sensory modalities in other species, is to make sure that male most of the time behave like males and female most of the time behave like females. Now, interestingly, the bisexual potential of the brain was demonstrated already very convincingly in many non-mammalian species, in particular in fish, in reptiles, in flies. So this seemed to be a very conserved part of the brain. So what we would like to know is, how does that work? And more specifically also, we demonstrated this for females, but for parallel, for consistency, our model postulates that there must also exist some female typical circuit in the male brain. And I'll come back to this in a little while. But first, let's try to investigate this area here, this control area, this switch that enable the animal to control whether it will behave like a male or whether it will behave like a female. Is there a brain area somewhere that could be in which we could see the emergence of some sex-specific computation? And the area that we analyzed was more specifically the medial amygdala. And the way we came into this is to look at how different the voronasal detection, the computation in the accessory olfactory bulb, and then the computation of the medial amygdala differ between males and females. So regarding the voronasal organ, a very talented postdoc in my lab, Yo Isogai, several years ago, developed a molecular toolkit to associate specific social cues with specific set of hormonal receptors. I'm not going to develop or explain this into much detail, but basically this gene called EGR1 has the ability to be highly expressed when a set of neurons is activated. And so the idea is to have a mouse interacting with a specific set of cues, and then we sacrifice this animal, section the hormonal organ, and look for the expression of this gene, EGR1. And whatever neurons express EGR1 on high level are neurons that have been highly activated in the behavior preceding the experiment. And so the behavior could be the interaction with a male, with a female, with a predator, with pups, with all sorts of cues. And in addition to the readout of EGR1, we also look for the expression of specific hormonal receptors. And if we see a particular set of neurons both expressing EGR1 and a particular hormonal receptors, then we can deduce that that receptor has been activated by either female cues or male cues or predator cues. And so Yo did this with a very large number of receptors, over 100. And what he found was a very large number of receptors, actually the majority of the hormonal receptors were activated by heterospecific cues, in other words, predators. And then a very small subset were activated by either male cues or female cues. And interestingly, you can see that there is almost no overlap between the receptors that detect males or females or mouse cues and non-mouse cues. In other words, these receptors have a very high specificity. We also didn't find any difference between the specificity in males and the specificity in, of female detection. In another set of experiments, we now recorded from the accessory olfactory bulb and the medial amygdala. And so here are the result of this recording experiment. And what each individual dot represent here is the specificity of a particular neuron, a particular unit that was recorded for either predator cues, male cues, or female cues. So I think this triangle is quite intuitive. If you have a unit placed here in this part of the triangle, it signifies that this unit is very specifically activated by male cues. If it's here by predator cues, if it's here by female cues, and somewhere in the middle by a mix of predator, male, and females. So that's what we found in the accessory olfactory bulb, and this is what we found when we recorded from the medial amygdala. Now there's something quite different in these two representations. You can see that the majority of the units in the accessory olfactory bulb are located in this central triangle, suggesting that these neurons are actually computing or integrating signals from both predators, males, and females. So this is quite different from what we observe at the receptor level. In contrast, in the medial amygdala, it seems like the units are much more specific, which is that the majority of the units are now found in the different poles of the triangle, meaning they are more specific to predators or males or females. So what is this transformation trying to do? Well, we think that one possibility is that this integration part here enable the animal to generalize across different types of predators. So maybe it doesn't really matter that the mouse is detecting a fox or a snake or an owl. These are all predators, dangerous cues, and the animal has to flee. And so this is what the accessory olfactory bulb is trying to do, is to integrate multiple signals and then re-dissect them in various categories. So that's one possibility. But somehow, something much more interesting emerged from these different recordings. And that is when we now looked at these plots according to whether we recorded from a male brain or a female brain. And when we do this, here are the data. So the red dots are units that were recorded from a female brain, and the blue dots, units recorded from a male brain. And you can see that in the accessory olfactory bulb, more or less, the blue and the red dots are intermingled. However, in the medial amygdala, you can see that the two sets of blue and red dots are completely distinct. And interestingly, if you look, for example, at the red dots, so recording from female mice, female brain, you can see that many of them are sensitive to predators or are sensitive to male cues, but almost none are sensitive to female cues. So in other words, these female are sensitive to predators and cues from the other sex. Similarly, if you look at the blue units, they are sensitive to female cues and predators, but very few are sensitive to male cues. So there is a very clear sexual dimorphism here. So there is a very interesting transformation here in which from a gender-neutral computation, now we obtain this emerging property of a sex-specific computation in the medial amygdala. Now, interestingly, we looked at what's happening in the juvenile medial amygdala, meaning right before sexual maturation, right before puberty. And what we see is that this sex-specific computation doesn't exist yet. So this is really something emerging later. During the peak of hormones that are released at the time of puberty. Now, obviously, one would expect steroids to be particularly important, and indeed, we looked at two types of mutants. One in which is an aromatase knockout. Aromatase is an enzyme that transforms testosterone into estrogen. And it turns out that estrogen is actually the main molecule that masculinizes the brain, the young brain. And as you can see, in the aromatase knockout, now these light blue dots are switched a little bit more towards the female type computation. So aromatase indeed is essential to help masculinize the brain and masculinize the medial amygdala. Similarly, if we inject estrogen into females, remember I just said that estrogen is the main hormone that masculinizes the brain. And normally young females do not have access to estrogen. But if you inject this in young females, you can see now estrogen-treated animals have dots that are switched toward a more male-like type of computation. So it seems that sex steroids are essential to the emergence of this computation. And this transformation of the circuit really appears only at puberty, when in fact this organizational effect of steroids were predicted to be way earlier than this. So it might be that hormones act very early, but at least the manifestation of this sex specificity really appear only later. So what's happening here? Well, we don't know. We are working on this, but it's possible that sex hormones enable a specific cell death that lead to this very sex-specific computation. It's possible that there is a synaptic refinement, so pruning of the wrong connections. It's also possible that the local circuits, so interneurons, are now functioning and strengthening the circuit that correspond to the detection of the cues from the other sex. Now, more recently, and actually in a work supported by the Simons Foundation, we were interested to know how the so-called social peptides like oxytocin act upon this sex-specific computation. And the reason is oxytocin has been shown already several years ago to have an important function on modulating the activity of neurons in the medial amygdala of the mice. And because we just discovered that this medial amygdala has this really interesting sex-specific computation, we were curious to know what would oxytocin do on the type of phenomenon that we had just develop. And I don't have to tell this audience, oxytocin has been identified as being a peptide important in many social interaction, parental interaction, bonding, and even trust building or other maybe more debatable function in humans. So Chen-Chin Yao in the lab decided to look at social interaction in oxytocin mutant. And in particular, she used this 3-chamber test in which a male mouse is put in a center chamber and then just left to wander in different areas. In that case, in this first control test, these other 2 chambers are empty, but then in the specific social interaction test, there is a female and a male that are each put under a cup, and the cup has many holes, so the male can interact with the male and the female, touch and smell them, but actually neither attack nor mate with them. So what's happening here? Well, if you look at the time spent by this male in the various parts of these chambers, you can see that if the cups are empty, the animals spend equal time on one side or the other. But if there is a male and a female, here this tested male will clearly prefer the chamber in which the female is present. Now, interestingly, if we now look at the male mutant for the oxytocin gene, now we can see that this preference for the female side completely disappears. Now, we then tested female mice, both mutant and wild type, and here we absolutely don't see first any preference in the wild type females. In other words, the female goes towards the male or the female intruder in equal proportion, and there is absolutely no phenotype observed. So this is very interesting because it shows that oxytocin has a role in establishing the social preference of male mice towards females, and that this role is actually sexual dimorphic, which is it's observed in male only and not in females. Now, we tested this male more extensively, and we found that these oxytocin mutants have a normal detection of odors. they have a normal detection of predators. So it seems like the defect we observe are really a defect towards social cues very specifically. We also observe exactly the same phenotype if this time we use a different type of mutant in which we delete the oxytocin receptor from the medial amygdala, from these areas that we suspect is really the center of the sex-specific computation in the mouse brain. We found this if we delete the oxytocin receptor from the medial amygdala, but not from an adjacent area also involved in social interaction called the bed nucleus of the stria terminalis. We also found this phenotype with the oxytocin receptor mutant, but not with the vasopressin receptor mutant. And this is important because oxytocin is actually equally able to interact with the oxytocin receptor than the vasopressin receptor. So that's really point to the oxytocin receptor as being important. Moreover, we are able to rescue the phenotype in the oxytocin receptor knockout by ectopically injecting a virus expressing the oxytocin receptor in the medial amygdala, but not in the bed nucleus of the stria terminalis. So there are two important aspects of this experiment. First, it's an additional control that the medial amygdala is essential for the phenotype we observed, but also it shows that the phenotype can be rescued in the adult. Therefore, oxytocin acts not as a developmental molecule, but rather as a molecule that constitutively modulates the social interaction of male mice towards females. So again, oxytocin signaling in the medial amygdala through the oxytocin receptor is essential for the social preference of male mice. So then we turn to Joe Bergen, who had done this recording in the medial amygdala, and asked, well, if the behavior of the animal in the oxytocin mutant is different towards males and female, how does that translate in terms of neuronal activity? So Joe performed a series of experiments in which he recorded from the medial amygdala while activating the warm nasal organ with male cues, female cues, and predator cues. And so here, for example, you can see the recording of a particular unit that is firing in response to female cues, female being represented in red. So in the control animal, so this is a Cre- animal that is crossed to a fluxed mutant, so conditional mutant. So this is a wild-type control. You can see that the units recorded are either sensitive to females or predators, and as expected, very little respond to male cues. But if we now ablate the oxytocin receptor in a Cre-positive mouse, cross to this conditional mutant, you can see that now the type of recording is very different. We have fewer units responding to females, and now we see emerging units that respond to males. So in other words, here in the control, we have a preference for the opposite sex cues, and this preference is abolished when we remove the ability of the medial amygdala to detect oxytocin signal. And this is shown here. If we look at the percentage of units that respond to female cues, male cues, or predator cues in the control versus the mutant, we can see a drop, a very significant drop in the number of units responding to females, a small increase, but significant, in the number of units responding to males, and then a little increase of the units responding to predators, but the overall strength of the response is more or less the same. So this is very interesting because it suggests that the oxytocin signaling in the medial amygdala modulates the response, the neuronal response of specific social stimuli. And very specifically, it's essential to promote the response to females and also to diminish the response to male cues. So we did another experiment to just see how reversible or how constitutive this function was by injecting into the animal, by IP injection, antagonist of oxytocin. And as you can see, this is the type of cartoon I showed before. Male has a normal preference towards the side of the cage that has female. But if we inject an oxytocin receptor antagonist, now this preference is abolished. And similarly, if we now look at the cartoon of the neuronal response, we see that the preference for females observed in the wild type now is abolished after injecting the animal with oxytocin receptor antagonist. And I would like to show you really the type of raw data that we obtained, because I think they are very striking. This is before the injection of the drug. You see the recording of this unit that is sensitive both to female cues and a little bit of predators. And after you inject the oxytocin antagonist, suddenly the response to conspecific females completely disappears, but the one to predators is unchanged. So there is something really amazingly specific of the action of oxytocin and oxytocin antagonists in modulating the response to conspecific. So again, OT regulation of social preference in the medial amygdala of male mice is constitutive in the adult and not developmental regulated. We also were able to trace the origin of oxytocin by, I won't go into very much detail, but basically we injected in an oxytocin Cree mouse a virus that enabled to follow the projection of these oxytocin neurons and see whether we can see fibers in the medial amygdala. There are two main areas in the hypothalamus that generate oxytocin. One is in an area called the paraventricular nucleus of PVN, and the other one is the supraoptic nucleus, or SON. When we inject the PVN with this virus, we can see a lot of green fibers in the medial amygdala, but we see very few when we inject this other area, right here, called the SON. And so this was important because in the next set of experiments, we decided to specifically activate the oxytocin-making neurons in the PVN, because we knew the origin of oxytocin likely came from these neurons. And so we use for this an artificial receptor that is specifically activated by an artificial ligand called CNO. So the idea is just to trigger the release of oxytocin at will by injecting these genetically modified mice with this ligand called CNO that otherwise wouldn't do anything to the mouse. So our infection with the virus expressing this artificial receptor was very efficient. And when we stimulate oxytocin release, we see this time exactly the opposite action as we had seen with the antagonist of the oxytocin receptor, which is an increase in the response to female cues. And here's the raw data for one of the units. You see this unit is very silent to both female cues, male cues, predators, ringer. And when we inject, when we stimulate with CNO, then we suddenly see the emergence of this response to females. So, these, I think, these findings are really interesting because they show they identify a role that had never been identified so far for oxytocin in enabling social responses, in this case only in males and not in females, so in a sexually dimorphic manner. And I think what is very striking to me is that, you know, oxytocin and other neuropeptides are often described as neuromodulators. But here, this is more than just a modulation. I think there's something just very profound in the effect of this peptide is really enabling the response of a male vis-a-vis a female. And if you remove oxytocin, there's simply absolutely no preference for male versus female cues. So again, a strength of oxytocin function that we really didn't really expect. So to conclude this part, I think that we really showed that the medial amygdala has a really important part in enabling this sex-specific computation, which is an essential part of the control of social behavior in the mouse. Now, as I mentioned, our model postulates that the male and the female brain have components of behavior circuit of both sexes, and that the voronasal system has this essential role in enabling the gender specificity of behavior. But the experiment I've shown you so far really related to females that seem to behave like males when we remove the voronasal system. And so an essential question is, do we see something similar in males? In other words, if we observe a male that is deprived of voronasal function, do we see some ectopic female behavior emerging? And this set of experiments is what I will describe today, right now. And the female-specific behavior that we decide to observe is maternal behavior. So it's very well known that in mammals in particular, females have the primary role in nursing and protecting infants. It makes a lot of sense since mammals have enabled the development of the fetuses in utero for a long period of time, and then lactate the infants for also often long period of time. So mom really has a primary interest in the survival of the infants. And occasionally, males also participate in raising infants. But actually, in the animal kingdom, this is very rare. And instead, what is observed most of the time is a very different behavior, which is infanticide. And this, surprisingly, has been discovered relatively recently, in the late 70s, by a graduate student in the psychology department at Harvard called Sarah Hardy. She was observing this species of primates, the langurs, in India, and she observed that each time foreign males, intruder males, were entering a particular group, then all the infants were either disappeared or were found wounded. And she was the first one to elaborate on this behavior and describe that these intruder males were invading, finding these females that were taking care of the infants, and in order to gain access to the females that were not receptive otherwise, kill the infants, and then sire their own offspring with these females that were now had become receptive. And so this actually caused an enormous uproar. Nobody believed that when she discovered it, because people looked at primates as a proxy to look at the evolution of human behavior. And obviously, if primates do that, then who knows what human could do. So there was a total denial of this is occurring. But then people start to pay attention. And they indeed found infanticidal behavior in many primates, most primates, in most mammals, and in fact in most animal species to start with. And so she proposed this evolutionary explanation of the access of the male towards female by killing their infants. And interestingly, this obviously is very costly, you know, to just kill infants. So evolutionary, there is a response to infanticide, and that is likely to be monogamy. So monogamy, if a male and a female have stable relationship, and now both the male and the female participate in raising the infant, there is no need for infanticide. So there is no need to sacrifice the life of infants, and then so that's an interesting consideration. So Herbert Wu, who is somewhere here, and really started this work, first tested whether in mice in which infanticide is also observed, then suddenly this male could be somehow parental, in other words, maternal somehow, if one would remove the function of the voronasal organ. In other words, is the voronasal organ in males also inhibiting this male-specific behavior that is parental behavior. So male mice, virgin males, are spontaneously infanticidal, and when they are put in presence with pups, they immediately attack them and kill them. So what you have here is a virgin male that is deficient for voronasal signaling. And what we want to know is whether this male is still infanticidal or somehow has acquired some female-like characteristic. So here's this male. Again, it's a virgin male that is deficient for VNO signaling. And interestingly, you see it already does something surprising, which is it's building a nest, which otherwise males wouldn't do. And you have some pups here. So it's approaching the pups, grabbing a pup, bringing it to the nest, and the video is going to stop here. But in the next few minutes, this male is going to go and retrieve the pups one by one, and then crouch around the pups exactly like mom would do. In fact, you would look at this video, you wouldn't distinguish this male mutant from a normal female taking care of her pups. So this is the quantification of the behavior. This is a virgin control, virgin male control. The majority of them are infanticidal, and in contrast, the virgin females are all parental. In the TRPC2 mutant, now none of the male, literally, or very few, stay infanticidal, and instead they either ignore the pups or are parental. And that's what is found also in control females. Now, interestingly, these males really do, these mutant males do everything that mom does. They retrieve the pups, they groom them, they build nests, they huddle with the pups in the nest. They do everything. It's just that they are a little bit more slow in being dads, but that's about it. And obviously, they can't nurse. So clearly, I think this experiment suggests that in the male brain somehow, there's the capacity to be maternal. And so this is shown obviously as a drug on humans, but regarding mice, you know, one might wonder why in these virgin males that are normally infanticidal, why are these males inhibiting a circuit such as maternal behavior? Why is the circuit underlying maternal behavior still existing in the male brain if these males are normally infanticidal? And you know, it's interesting to look across evolution, and you know, whether you look at insect, fish, frogs, reptile, birds, or mice, they are a species in which mom takes care of everything. They are also very similar species in which both the male and the female take care of the progeny. And that's true also for insect, fish, frogs, reptile, birds, and mice. And they are also species that are very similar, in which the male this time takes care primarily of the infants. And so it's interesting that these species are so similar to each other, because it really suggests that these brains are likely to be very similar, but somehow the ability of taking care of the progeny or not is either expressing itself or being inhibited. So this is quite interesting. So, so why would the male have a maternal circuit? Well, Herbert, at the beginning of his project, came to me with a paper from this group, von Saal, who had an explanation for this. And what he proposed is that actually males are normally paternal after they mate with a female. So they have a switch in their behavior from infanticidal to become paternal. So that seemed really interesting. But we were very skeptical because of the proposed timing. What von Saal was proposing is that they do not switch to become paternal right away after mating, but 3 weeks after mating with a female. Now, 3 weeks is exactly the gestation time in mice. So you have to argue that somehow the male who mates with a female, nothing is happening for 3 weeks, and then suddenly, at exactly the same time as the pups are born, now this male is no longer infanticidal, he's becoming paternal. And somehow, it seems to be too good to be true. So Herbert decided to confirm this experiment. And amazingly, actually, von Zal was absolutely correct. So he put some male in contact with females and then tested them immediately after mating and at time 0, and they were all infanticidal. He tested them 1 week to 10 days after mating with a female, and they were also all infanticidal. Around the time of the birth of pups, and half were parental, half were ignoring, but none were infanticidal. And then 1 week after the birth, they were all parental. So that timing seemed to be correct. And this is the control in which males have never been exposed to females, and they are all infanticidal. So in this experiment, actually, the male and the female stay together. So the switch of behavior could be due to the cohabitation of the males and the females, or there could be some type of timing occurring from the precise moment of mating. So we want to test what was important. And what we did was to separate males after mating with a female, and then put them in isolation, and then wait till 3 or 4 weeks, and then test them towards pups. So these animals have been removed from the female immediately after mating. These have been kept in presence of female for 9 days, 18 days, 24 days. And when we look then at the number of mice that are infanticidal in red, or parental in blue, or ignoring in yellow, you can see that the longer the male and the female stay together, the least infanticidal the animal is, and the more parental they are. So clearly, the contact with the female leads to more parental behavior. But for me, what is really striking is that an animal, a male that has been removed immediately after mating with a female, half of them are parental when you test them 3 weeks later. So somehow, mating with a female is sufficient to trigger the switch in behavior 3 weeks later. So this is to show you, this is a certified infanticidal male that has been tested 3 weeks after mating with a female. And now it has built a nest, retrieving the pups to the nest, one by one. Does a very good job, you'll see. Now it has them all. It's a bit slow. Now making sure nobody is left. Yeah, everyone is here. Let's go and crouch with them. So really good dad. So in the last few minutes, then let me go into the secret underlying this. So the idea is that in the wild, there is a dominant male with a group of females. This male is constantly mating with the females. The female have communal nest. So this male is paternal. It's paternal for a long period of time, as long as it's dominant, as long as it has access to the females. But these subordinate males don't have access to the females, so they are constitutively infanticidal until they kick out the dominant male and mate with the female. So at the neuronal level, it seems that there are really two brain areas, one that would trigger infanticidal behavior, that would be the default pathway in virgin males, and another area that is the default pathway in females, but also used by dads, and that's the neurons controlling parental behavior. So Herbert decided to search for these neurons. And so what he did was to design an assay based on the increase in CFOS activation in areas of the brain that are involved in parental behavior. So the assay is to have either fathers or virgin female interacting with pups. Then he killed the animal half an hour after the social interaction, and then looked for neurons that were overexpressing CFOS as a proxy as a marker for neurons that had been activated during the social interaction. And he found one area in particular that it was very highly activated, and that's the medial preoptic area. Now, this is not exactly surprising. It has been found already over 50 years ago. It was known that the medial prefrontal area was essential for maternal behavior. The key, however, is to know which are these neurons. What is the identity of these neurons? And by trial and error, Herbert was able to identify this particular peptide called galanin as being nicely overlapping in expression with the neurons expressing CFOs. So in other words, the idea is that a large percentage, a large fraction of the CFOs positive cells activate during parental behavior, both in males and females, express the peptide galanin. Now, galanin does a lot of things. It's expressed throughout the brain. It's involved in nociception, sleep, feeding, thermoregulation, osmotic regulation, reproduction, lactation, so a lot of different things. So we're not sure galanin does anything to parental behavior per se, but it's a marker in the preoptic area for neurons that might be involved in infanticidal behavior. So Herbert used now a transgenic mouse that expressed the Cre recombinase under the control of the galanin promoter, and in a first set of experiment injected a conditional virus expressing a toxin, the diphtheria toxin, and that enabled to specifically ablate neurons expressing galanin in the preoptic area. And when he did the experiment, he found that if over 50% of the neurons, galanin neurons, are killed, so if the number of remaining galanin neurons is below 50%, then virgin females lose their maternal behavior and instead become infanticidal. If he tested now the experiment, did the same experiment in fathers and mothers, he found that also here, when over 50% of the galanin neurons were killed, now these animals lose their parental behavior. Interestingly, in that case, they do not switch to infanticide. And we think that the reason is mating has somehow inhibited constitutively neurons driving infanticide. So if you now kill galanin neurons, you wipe out the parentic behavior, but you still don't activate neurons involved in infanticidal behavior. However, in virgin females, this is very interesting because it seems that the galanin neurons both promote parenting behavior, but also inhibit infanticidal behavior. So MPOE galanin neurons seem to be essential to trigger parental behavior in both males and females and to inhibit infanticide. Now, what about the other experiment, which is, can we take an infanticidal male, activate galanin neurons, and see what's happening? And so using optogenetics, Herbert performed this experiment. He took virgin males, injected a virus expressing channel rhodopsin in galanin neurons, inserted an optic probe, and then each time the virgin male, so the infanticidal male, approached a pup and the snout touched the pup, either he did nothing or he activated these neurons by optogenetics. And so I think the beauty here is that the same animal can be, in random assays, either has activated galanin neurons or not activated galanin neurons. So these are male tested without galanin neuron activation. And you can see the assays have to be stopped very quickly because these animals are extremely aggressive, which is shown here by this red dot. So every line represents a different behavioral assay from a different animal, and red represents the episode of aggression. The same males, exactly the same males, when these galanin neurons are activated optogenetically, now you see disappearance of attack, and instead this long episode of grooming behavior. So this is very interesting because it shows that these galanin neurons, the activity of these neurons, is sufficient to inhibit infanticide and promote parental behavior. Now, let's go back for a minute to these dads that are almost as good as moms, but just a little bit more slow. We thought, well, maybe if we take a dad and activate galanin neurons, maybe that would make them better dads. So we took these fathers and activate galanin neurons when they were in presence of females. And what you can see before stimulation and after stimulation. So before stimulation, there's a little bit of grooming, but not much. There's a lot of crouching, which is this episode in green. But when you stimulate galanin neurons, now they are grooming, they increase their level of grooming behavior very extensively. So I found this experiment very funny because what's happening here is that, the males are crouching with the pups, it's like they are sitting on the sofa, eating popcorns, watching TV, and then suddenly you activate galanineons and now they feed the pups and they make them bed, they go and take the shower, and, they become just very active dads suddenly. So, maybe there are different styles of parenthood, maybe mothers are more active and fathers, they just like to sit down and be a little bit more passive. Or alternatively, there might be different ways to activate galanineons, and that lead to different types of behavior. And so this is a very interesting hypothesis because, parenting behavior is a lot of different things. It's building a nest, it's retrieving the pups, it's grooming the pups, it's crouching with them. So what regulates all these different behaviors? Maybe it's different population of galanineons that do different things. And so Herbert has an experiment that unfortunately are not finished, tried to address this by recording galanineons in an awake behaving females here in presence of a pup. And what you can see is that this particular unit seemed to be firing when the female is retrieving the pups. And in subsequent experiment, Herbert was able to find that there is diversity in galanineons. Some are activated when animals are retrieving the pups or building nests. Some are activated when the animal is grooming or retrieving. So it's possible that there is a diversity of galanineons that each do different things. I'm a bit running out of time, so just in the next two slides, let me just briefly mention where we are at now. We want to understand how the control of parental behavior is working. These neurons are essential, but how do they transmit the order to the brain to do the various steps that constitute parental behavior? So Johnny is trying to identify both the input to galanineons, what are the brain areas sending information to galanineons, and also the output. So what are the projections? What are the input to these galanineons? And I'm not going to detail any of the neurons, any of the results, because we don't understand them yet. What we found is that there are about 18 different areas that receive projection from galanineons, and we can attribute some function potentially to some of them, just based on what these brain areas are. Some might be motivational or sensitive to hormones. Some might regulate the motor aspect to parenting behavior. Some might be involved in inhibiting aggression. If we look at the input, we also find 18 to 20 different input areas. And also here, we can try to decompose in various functions. Interestingly, many of the projection areas are also found in the input area. So they are likely to be a lot of recurrent circuits that somehow help modulate the system. But how does that work? This we don't know yet, and hopefully we'll be able to understand. But there is also a very different component to this circuit, and that's the hormonal regulation of parental behavior. And interestingly, a former postdoc in the lab, Tali Kimchi, identified the AVPV, which we found as being both an input and now output of galanine neurons, being also essential in females in maternal behavior. And these neurons are themselves regulating the oxytocin release from the paraventricular, the paraventricular nucleus. Now, why this is important? Well, because hormone levels change enormously in females during pregnancy and at the end of pregnancy. There is literally an enormous drop in the release of some hormones, so progesterone and estrogen really drop. There is an increase in other hormones, prolactin and oxytocin in particular, and those really are preparing the body of the female for parenting. But these hormones are not the whole thing, because males, as we've seen, are able to be parental, and also foster parents can be also equally nurturing. So how is the hormone working? We don't know. And then finally, there's something really interesting about the role of hormones and how these changes in hormones can affect the quality of parenting behavior. And I would like to just mention that as my last slide, that there's something profound happening at childbirth in women. And you can see here the enormous increase in psychiatric hospitalization that occur right after childbirth. And this is caused by probably many factors, but one of them is the sort of hormonal chaos that is occurring right at childbirth, with these extremely potent hormones that have their level dropping or increasing in very dramatic way. And postpartum depression is a really very prevalent disease of young mothers and also fathers. So just to finish, I think that we've uncovered what I think is the first molecular and cellular elements of infant evoked behavior circuit that are both present in males and female brains, and that are regulated by social experience and physiological status. We're now working on the other aspect, infanticidal behavior, as well as the switch between the two behavior. We think that this is a really interesting paradigm to look at instinctive behavior, instinctive social behavior, and their plasticity. And then finally, it is thought that the circuit that underlies parental behavior is likely to be the foundation upon which other types of social bonds have evolved. So whatever we discover here might be more applicable to other types of social interaction. And let me thank the hero of this work. Herbert really did the lion's share here of most of the work on parental behavior that I discussed, together with Anita, Autry, Chen-Chin Yao, and Joe Bergen did the experiment in the medial amygdala, and Johnny Cole is doing some of the circuit analysis. Thank you very much.